Doron Levy, who is giving the first plenary lecture. I'm sure you all know Doron, very well known in mathematical biology. He's at the University of Maryland, where he's the chair of the mathematics department. He's also the director of the Brin Mathematics Research Center. And according to his Wikipedia page, besides mathematical modeling, he also does magic and is a magician. So today we'll be talking about the magic of fighting drug resistance with math.
Okay. Okay, now that he's gone, we can have some fun. Okay, good. Okay, so thank you so much, Dominic, for the introduction. It's great to be back to the Fields Institute. And since this is the first talk of the workshop, what I thought of doing is like taking it easy and just showing you some things about drug resistance and cancer. Do not show that again. How do we get rid of that? I do not know. Let's try to, here, got it. Okay. This is better. Now even I can see the slides. Okay, the key point is that this is a joint work with, what I'll tell you is about joint work with multiple people. And in particular, I'd like to point at Michael Gottesman, who was the deputy director of the NIH in charge of the intramural research there, but also the head of the laboratory of cell biology. And strangely enough, for a medical doctor, a big believer in math and the impact that it can bring. I even seen him once giving a talk at the NIH with integrals and equations, and this was really inspiring. But even more so, it really is a question of how can we make an impact, and what kind of questions can math answer that experimentalists and clinicians may not be able to do. Yeah, I said, well, okay, I come from the University of Maryland, so might as well just have a small advertisement for the university. So this is Washington, D.C. here, and this is where the university is located. That's this star. This star here is where the National Institutes of Health, the main campus in Bethesda, is located. So is the Walter Reed Military Hospital. This beltway, the radius is about 10 miles, or in kilometers, that makes it 16. So you get the scale. This is about half an hour drive, and that actually is what it takes if you know how to avoid the traffic, which is very difficult, of course. This is the math department at the University of Maryland, and this is a beautiful view of campus. Okay, now with this being said, let's move on to the talk. Drug resistance in cancer. I want to tell you a little bit about that and why math may be important in looking at drug resistance in cancer. Where does this resistance come from? But the focus of this talk really is on heterogeneity and resistance. So this is my favorite slide. It comes from a paper by Gilles Gottesman from 2010, in which what you see here is a caricature of basically a sketch of a cell. So here you have the membrane. This is the nucleus of the cell. Here is the DNA. And what is prescribed here, described are different mechanisms of drug resistance. So, we can think about drug resistance as something that, the cell has evolved to be resistant to. But what does it actually mean? Okay, and does a cell have to do anything to be resistant to a drug? So think about what the drug has to do in order to act on a cell. Now, of course, there are many different kinds of drugs, and there are many different things that it may try to do to a cell. And of course, there are many different things that a cell may try to do to avoid reacting to the drug. So for example, the first thing that a lot of drugs have to do is to penetrate the cell and get into, to do whatever they have to. Of course, you can send signals from the outside, but sometimes it's more effective to go inside and do your work from the inside. For example, if the goal of the drug or the way that the drug works is to do something to the DNA, say to destroy the DNA or to damage the DNA, what the drug has to do is to get it through the membrane and then into the nucleus and then do something to the DNA. Now, there are many things along the way that can go wrong with the drug not being able to do what it has to do, what it's supposed to do. For example, the drug enters the membrane of the cell, and then, as we know, the cells have these mechanisms, these transporters, the efflux transporters that would then pump the drug out of the cell, right? So the drug tries to get into the cell, and it's being pumped out of the cell. Now, you'll say, why shouldn't we disable these mechanisms? Well, you can do that, but there's a reason why these mechanisms are there. There are all sorts of things that are supposed to get out of the cells and can't just get stuck there. So it's not necessarily a good idea to go and block these mechanisms, but you can try. In any event, even if the drug entered the cell, it can then be engulfed and metabolized. It can go into the nucleus and damage the DNA, but there are DNA repair mechanisms. It can send apoptotic signals, but there are anti-apoptotic signals, and so on and so forth. So what that picture really demonstrates is that when we are talking about drug resistance, we're talking about the very, very complex system. It's not simple. So why math? Well, this was the year 2012, where together with Levy and Michael Gottesman, we wrote a paper on the dynamics of drug resistance and mathematical perspective. What was interesting about this paper was two things. One is there was not a single equation in the paper. So in some sense, for me as a mathematician, this was the most difficult paper ever to write. Okay, that's one thing. The other thing is that journal where it was published has nothing to do with math, drug resistance updates. And it is a journal that pretty much has experimental work in it. And the question is, why was that paper there? And the answer is, well, this paper was an attempt to explain to clinicians and experimentalists what kind of questions can mathematicians ask, and in what way can we potentially help address the issues that come in the area of drug resistance. And this is an example. I mean, what I listed here are 3 questions. There are many more in that paper. And these are the classical questions. And there's a lot of work that was done in answering these questions. But really, let's look at these questions, for instance. So the optimal protocol for drug scheduling in terms of dose and timing. Well, pretty much everything that's done in the clinic is based on either clinical trials that were done for other individuals, or than those that are being treated, or on guesswork. Okay, so once a drug is approved, in principle, doctors can go and do whatever they want to, with certain limits, but I mean, what do they do? And how would they know what to do? Well, they don't have any quantitative tools to do that. And that's a place where math can enter and assist. Should you administer the drug through, continuously, or short pulses, or intermediate administration? If you have several drugs, should you use them at once, or should you use them, sequentially? I mean, the logic that people use in the clinic is not always the correct one. Sometimes doctors would use drug after drug after drug, and you ask them, why do you do that? Say, because when the first one fails, at least we'll have something else to use. Okay, and when the second one fails, at least we'll have a third one to use. But maybe that's not the best you can do. Maybe the best you can do is to use all the drugs at the same time or a subset of them. And when they fail, maybe there's nothing you can do, but it would still be better than doing things one by one. These are tough questions, but that's actually what clinicians deal with using some guidelines, but not that many quantitative tools. And that's where we try to bring in math. So as I said, the theme of this talk will be tumor heterogeneity and drug resistance. So what is tumor heterogeneity? Well, let's think of the following experiment where you look at cells and you sequence their genome or any other thing that you want to look at, okay, in the cells. I don't care what, mechanistic properties, some other kind of omics, whatever. And what you're looking at as what is the result of whatever you're measuring at different locations within the tumor. So you do that thing, and the result is typically you will see different things. And the conclusion is that tumors are heterogeneous. So what does it mean? Well, it means different things to different people and in different contexts. And we'll hear lots of talks that go in this direction where heterogeneity is studied and throughout this workshop. But for us right now, all that it means to me is maybe there is going to be a heterogeneity in response to drugs. Now, I said genome. I just want to emphasize that the genome is not everything, right? And that's very important to understand. There's way more to the dynamics than what you see on the level of the genome of an individual cell. So that goes to the whole story of genetics versus epigenetics. And again, we'll hear a lot about that during this week. But as an example, just to emphasize that point, I want to show you results, experimental results that come from a group. So AMIGOS refers to an acronym that was coined by the National Cancer Institute, and that is Applied Mathematics and Germinating Oncology Solutions. That's the AMIGOS. Okay. What you see here are just experiments. These are 3D cells, triple-negative breast cancer cells that were grown in the Peyton lab. And what you see is a span of 48 hours. And each line represents a single cell lineage. And they're all identical. These are identical cells. And they all behave differently. And they're in the same place, same conditions, same everything. And they behave differently. And what do I mean by behave differently? Whenever you see something splitting, that means that the cell divided. Whenever you see an X, small x, this red thing at the end, that one died. And you see that these cells here did not divide at all. These cells divided many, many times in a period of 48 hours, and everything behaves differently. So identical cells, identical everything, identical conditions, identical environment, different behavior. So we have to be very careful. And there are many other things that can happen. What does it mean that the cell is resistant to a drug? Let's say that the drug is supposed to affect the cell cycle, to slow down the mitosis phase or any other thing, okay? So what happens if the cell is not dividing? Well, it doesn't do anything in response to the drug. So typically in a given experiment, if you take, I don't know what, on a Petri dish, if you put cells and you just flood them with a drug, you will still have like maybe 15% or 20% or 10% of the cells surviving. They don't have to mutate to anything. They don't have to have anything inherent that says that they're going to be resistant to the drug. They just don't see the drug. They're not responding to the action of the drug because they're not dividing. Okay. And then drug is gone. They'll wake up. They'll start dividing. What happened? As if nothing happened. Okay. So what is the ultimate goal in these studies? In my mind, well, I don't know what the ultimate goal is, but let's play a thought experiment here. And I call this a science fiction ideas, okay? So number one, let's say we know precisely the initial data. That is, a tumor is diagnosed in the patient, And upon diagnosis, we assume that every cell may have a different response, a different level of resistance to a drug that is yet to be administered. But I know somehow, using technology we do not have today, how every cell will respond to the drug. We can't do that, but let's say we could. And let's take it one step further, and let's assume that we can fully control the treatment. That is, we can control how the drug gets to anywhere we want to in the tumor, in whatever dose we want to, at whatever time we want to. We can't do that, but let's assume we could. And on top of that, we can take it one step further and assume that we can fully and dynamically know how the tumor, how all these individual cells are responding to the treatment and adjust accordingly. Of course, we can't do 1 and 2. We certainly can't do 3. On the level of the cell, not, okay, here's a tumor, let's image it. And the question is, what is the best we can do? So remember, right now, patients are treated without knowing any of that stuff. But let's say, think like mathematicians for a second. Let's say we have everything. What's the best we can do? Of course, there's a question of, what do I mean by best? What do I mean by optimal? All right. So, I mean, I cannot say anything about drug resistance without going to the very beginning of these studies. So let me put several slides about modeling resistance. And, you know, if you, I mean, and most of you have seen this, bear with me. If not, that's a good opportunity to see it. Okay, so modeling resistance goes back to Luria and Delbruck. That's a work from 1943 for which they won the Nobel Prize in 1969. It's a combination of experimental work and mathematical work. It is a Nobel Prize also for math. And at the time, there were no antibiotics, so the setting was a bit different. And the picture says it. So when bacterial cultures are challenged with a phage virus, most of the bacteria is killed. But the cultures tend to grow, and when they do, they resist infection by the same phage. If you don't like phage, put now antibiotics. Okay, so you charge bacteria with antibiotics, you treat bacteria with antibiotics. After whatever survived, what is left emerges as resistant to the antibiotics. And the main question was, what is the source of resistance? And there are two possibilities to address that. One was to assume that resistance evolves by natural selection. So the mutations that confer resistance exist prior to the infections, while the other option, generally speaking, and of course there can be a combination of these, but that is the basic work. And by the way, if you have not read the original paper, I highly recommend to go and read it. This is the way papers were written in 1940s, which are significantly more beautiful than the way we write papers today. I mean, there are actual stories there, and it's nice. So the second way of thinking about it is resistance is acquired. In other words, it is something that comes as a result of the administration of the drug, or in this case, this challenge of the phage. Okay, and this whole work was set to decide whether it's natural selection that's prevalent there, or is it acquired resistance? That's a very, very difficult question to study, but how do you study it? And what they've done really is an interesting, very interesting experimental design. So the idea was take a small number of samples from the parental culture. Okay, so the likelihood of pre-existing resistance is small if you do that. If not, the whole thing fails. Okay, expand the culture to a fixed size, and then you can replace on selective media to determine the number of resistant cells. So you plate. Now, there are two options. I mean, if you had predetermined pre-treatment resistance, then this will show up. But since you start with so small pre-existing resistance, then you will not see uniform distribution when you plate. And you repeat this with many samples. So there's a difference that you will see in the variance. If resistance is inquired, that will only happen in the last step. If resistance is selection for pre-existence, it will happen along the way. And when you do that, at the end of the day, you look at the different plates with acquired resistance, you should see a similar distribution of resistance cells. With natural selection, you will see different distribution. And that's the experiment. And the conclusion was that what you see here is a natural selection. It's something that exists before the drug is administered. Okay, so this work, of course, is sufficiently more complex than what I'm describing here. This is the experimental component of it, purely set, but there's also an analytic component where they do study the variance. And from this variance studies, they infer the mutation rates and so on and so forth. So it's really a masterpiece, and it's always good to go back to the masters. Okay, that being said, let's go on. And what I want to do right now is, after convincing you that drug resistance is super complex as a system, I want to, just for a warm-up, show you the simplest possible model I can write. I mean, nothing is simpler than putting two populations and a deterministic equation and ordinary differential equation. Can't do any simpler than that. So what's going on here? Well, you have first a single drug, and I'm assuming an exponential cancer growth, and I'm assuming that the wild type cell, and two types of cells, there's a wild type cell and a resistance cell. That is it. What is resistance? Everything is inside resistance. Okay, and for me, the N will be the natural, the wild type cell, and R is the resistance cell. And the wild type cell has an exponential growth. So what we have here is just L and D are the birth rate and the death rate. So you have an effective growth rate. The same, and you can make it more complex, but the same growth rate and death rate for the resistance cell. And there's an increase in the population of the resistance cell that comes as a result of the division from the wild type cells. And this is a dynamics that you see before time t star. And the time t star is a time where we administer a drug. Before that, there is no drug. What happens after the drug? After the drug is administered, that adds a drug-induced death rate. The drug-induced death rate affects only the wild type cells, while the resistant cells still continue with their same dynamics. Can't think about something that's simpler than that. Yes?
Just a stupid technicality. Why don't you have a negative UN in the first?
Because what I'm thinking of, so the question was for those online, why is there a plus here and there's no minus to compensate for that? Because I'm thinking of whenever a cell is dividing, it does not change the, that when a cell is dividing, it's an asymmetric division where it keeps one copy of itself and creates one resistant cell. So it increases the population of resistant cells, and it does not change the population of the wild type. Now, I could have balanced things differently. This really is a question of the parameters, but that's a good question, and that is the explanation. Okay, and what one can do is, and again, that's the simplest possible thing I could think of. It's so simple that you can solve it analytically. I mean, there's nothing, I mean, these are really simple ODs. And you also think about initial conditions. The initial conditions will be that at time zero, there are no resistant cells. There is a certain number of wild type cells at time zero. And time t star is a time where the drug is administered. So one thing that, and you can take that now and do all sorts of things with it. I'm not writing down the calculations. I'm just writing the results. So one thing you can express is you can express the time of the treatment in terms of the number of cancer cells at the beginning of the treatment. N naught is the number of cancer cells at time zero. Capital M is the number of cancer cells at time t star. And given that, you can easily calculate the amount of resistant cells that are present when the treatment starts. Okay, so R at time t0. And one thing that you see here is that, I mean, unsurprisingly so, the number of resistant cells depends on all the parameters that I have here. So the mutation rate, the size of the tumor and diagnosis, what we started with, and the birth and the death rate. I mean, if you do the asymptotics, you can get rid of some of these parameters, but this is what happens at the time of diagnostics. If you do the 2-drug case, it's as simple as the 1-drug case. Effectively, you can, but again, I mean, this is very simplified. So you assume that a resistant population to 1 drug, resistant population to 2nd drug, a resistant population to both drugs, same dynamics before treatment and after treatment. You assume that all 3 populations are resistant to, are responding to the treatment, while the population that is resistant to both drugs does not. And actually, you get the same expression with a power 2 here. Okay, but why do I care about the amount of resistance present at the time of when the resistance starts? Because I can ask the question of what is more prevalent here. Is it the pre-treatment resistance or the resistance that develops during the treatment? And you can calculate these 2 things. And what you can conclude under reasonable mathematical assumptions is that the resistance that's pre-treatment resistance is greater than the resistance during treatment. Okay, so this is just a warm-up example. I try to convince you that drug resistance is significantly more complex than that. So let's move on. Let's try to, beef things up and add some heterogeneity into the picture. So the first thing that I'm going to do is I'm going to treat resistance level as a continuous parameter. There are various works in the literature that have done this. You do not have to look at it as a continuous parameter. You can do different levels of resistance and consider that as a discrete thing. You can associate the continuity with various things. I'll not get into details about that, but I will assume that resistance is a continuous parameter, where if you look at a tumor, there are different levels. This actually picture shows the discrete levels of resistance. So the yellow will be maybe the sensitive cells, the red will be those that are fully resistant, and the blue and the green will have some level of resistance. But think about all these cells having different levels of resistance. And now I want to look at a way of modeling using a resistance using a discrete level of resistance. So what do I see here? Well, my population of cancer cells, this N, is a function of 2 parameters, X and T. X in this case is not space. It will become space in a second. There's no space yet here. X is this trait. It's a level of resistance. Sensitive cells are assigned X equals 0. Resistant cells are assigned X equals 1. Level of resistance varies continuously between 0 and 1. What are the terms that we have here? How do they evolve? Well, different things can happen. One is they can expand. The other thing is they can die. There is a drug. This drug affects, as written, the birth term or death. Okay, so it's a negative term here for the drug. And they can also mutate. And the mutation here is a mutation with some mutation kernel, where what I'm allowing cells with one level of resistance to do is to convert to a different level of resistance. In fact, I can take this equation, I can, I'm sorry, sorry, I need to go backward, press a different arrow. Okay, sorry about that. One thing that you should notice here is that all these parameters are a function of rho of T. Rho is, it's not exactly the density because there's no space here. It's just the total number of cells that we have. Okay, so there's that. Now, there's a little bit redundancy in this system, the way it is written here. So you can rewrite it and rescale time and do all that stuff to simplify things and leave the dependence on this rho only in one term, where the others do not. But the birth here, yeah, okay, so that's it. Now, there are ways of looking at this thing, but instead of just telling you what that whole thing looks like, let me break it into small pieces. Let me take case number one, where I'm only looking at the trait-based growth. So I have here a birth, I have here the action of the drug, and I have a natural death. You'll ask me, why do I need to look at this at all? Well, remember, there is an X. So there is a dependence here on the trait. If there is no dependence on the trait, this is just y prime equals ay, okay? But there is a dependence on the trait. So every trait can behave differently here. And these parameters, okay, the growth, the natural death, and the drug also depend on the trait. Okay, so the dynamics, even on that simple system, with that simple system, can be interesting. Now, notice that I remove the dependence on the density. I call it density, this fake density. I'm adding it here. And for the third case, we'll look at the full model where mutations are also part of it. So the first case is a case that was studied by Alexander Lortz and Tommaso Lorenzi, Benoit Pertam, Jean-Claire Baugh. And what you see here is, and the equation is written here, a simple ODE, but there is this X, there is a trait. What you get is that the density, that's easy to show, goes to infinity as time goes to infinity. The ratio of n over rho goes to a collection of delta functions. So you get these spikes in the trait space. Okay, and that, of course, it depends on, you know, these functions. So here I plot several functions. The red curve is R, that's a growth. The blue curve is the death as a result of the drug, and the green curve is the natural death. Of course, if these figures were, if these graphs were different, then you know, you could get different things here. But in any event, what you see is a spike in the trait space. This is the sensitive cells here. These are the resistant. So this is the emergence of simple heterogeneity. Yes?
Can we ask questions?
We can ask questions, yes.
So the maximum of R minus E minus D could be attained in an interval, right? So that then the entire interval would go up.
Well, yeah, and that depends, of course, on these functions. Yeah.
It doesn't have to be
Yeah, of course. I mean, like I could take all these functions to be so that the whole thing is zero. I can get all sorts of weird things here. But yeah, you're right. Yeah, but this, there are conditions by which, yes, yeah, yeah. Try to be a little mathematically less technical. Let's look at what the second thing does, because that's a little bit more interesting. What happens when I add the density dependence? And when I add the density dependence, all of a sudden rho, instead of being unbounded, becomes bounded. Okay, so that's the main difference. And you still get these deltas, again, under certain conditions here on these parameters that I'm not writing down. And finally, if you do the whole thing with the mutation, the interesting thing that you get, not only the density is bounded, but instead of Dirac masses, you get these, structures that have some, more like what Thomas was asking about the interval. You no longer get these deltas. So this is really something that I like to think of more as a tumor heterogeneity. So you get the spread in this trait space. Okay. But this is very, very simple. This is, there is no space here. There is a density dependence in some strange way. There is mutations, but let's add space to the picture. Yes.
Can you give a little intuition as to if you go back to this slide?
Oops, I'm pressing the wrong button.
Tumor size is smaller, or you get more heterogeneity. So, I mean, intuitively, and I see it mathematically, right? But
the question is intuitively why?
Why would this selection mutation model with the density dependence give you just in some?
I mean, I think what it does is what the selection mutation does. The mutation think that this is basically, that's why I'm breaking it into the three pieces, right? So what the mutation does, it really allows to break out of this structure where you get that peak. That's the intuition. I mean, you get this concentration without the mutation. The mutation just allows things to escape.
And why the smaller tumor size? I mean, we're explaining this to a doctor, not to a mathematician. How would you explain it?
Why a smaller tumor size?
How would you explain it to a doctor? He doesn't know what
Yeah, I do not have a, no, yeah, maybe I need a doctor that knows integrals. Yeah, No, absolutely. I completely agree with you. Okay, let me think about that and we'll discuss it later, but that's a good question. I do not have an offhand explanation to that. Yeah, and yeah, okay. And I will say also that this is certainly, that part is an observational part. Okay, beyond, that's not the theorem. This is not the theorem, that part. This is the theorem, not this one. Okay. But let's discuss it later. Yeah. Because I do want to add space here. So, and I could have jumped directly to space, but I mean, space right now has the same elements as before. So we know that we can have density dependence, we can have mutations, we can have a continuous parameter. Let's look at what happens when you add space. Well, there's still a work of Alexander Lortz and the group there that they added, where they added space, only that they made space radially symmetric, using the elements that they previously had in the original model. Remember that when I had these three models, the first one, the one that did not have mutations and did not have density dependence, that's the one that corresponded to the original Lortz model, and that's the one that they are expanding here with this radially symmetric one. So again, I'm going to take it in steps. Their model basically has now three parameters. T is time, R is now space, and theta plays a role of what X played previously, that is a trait, okay? And what their cells do is they can give birth, they can die, and they can die as a result of a drug, cytotoxic drug. It effectively increases the death rate, okay? In addition, I mean, now we're adding space, so why not doing things a little bit more accurately? They add nutrients, and the nutrients impact, of course, the way that the cells grow, so the R term here. But that also gives an opportunity to introduce another type of drug, a cytostatic drug, which slows down the growth of the tumor. Okay, so now there are two drugs here. And for these two drugs, you can assume, I mean, there's space here, you can assume that they diffuse and there is a, you know, a source of these drugs. So there's a source and a consumption and the diffusion of the drugs. And that is the model. One thing that you will notice if you look carefully at this model, you will see that cells do not move. Okay, so there's space, but there is no motion. So, and the space is radially symmetric. So think about these rings that you have, and within a ring of radius r, there is a dynamics that happens, but the cell that is there cannot move to a different location. This is a model. And all the dynamics happens within these, you know, R kind of, yeah, rings. Okay, so why not allow cells to move? Okay, and along the way, let's add more things to the picture. So here's what we can take that model and adapt it in several ways. One is you can add density dependence to everything. Okay. As we've seen with the previous 1D model, the model with no space in it. The other thing, this is still radially symmetric. I'll break the radial symmetry. Don't worry. I mean, I'll do that soon, but let's start with that. The other thing I can do is I can add mutations. I know that that adds already, based on the previous model, something to the heterogeneity. And the third thing, along the way, might as well allow cell to diffuse in the radial direction, okay? So these are the three, and other than that, similar to things that, as we've seen before, we have nutrients, we have one drug that is a cytotoxic drug, a second drug that is a cytostatic drug that slows down the growth of the cell. And what happens? And then there is another story here about what are the actual uptake functions here. So there's a question of what does the growth look like? And that's a subtlety. It's an important one, but let me skip it. Okay, so there's a question of linear versus nonlinear. So this is what I want to show you. Okay, so if anything to remember, that's what I'm remembering for this talk, it's this one figure. So let me try to parse it, and let me look at the center graph. So what happens here, you have a picture, each picture like that corresponds to time on the x-axis, t-axis, and this would be the level of resistance. 0 is sensitive, 1 is fully resistant, on that scale of the trait. And the difference between this and that is just an increased dosage of the cytotoxic drug, okay? So what do you see here? What you see is that as time evolves, there's a gradual transition to more and more resistant cells. But that's not the most interesting thing in this figure. The most interesting thing in this figure is actually the very centered one. It's this one. What do you see here? What you see here is two things. One is it's a higher dosage of this cytotoxic drug. So compared with this figure, this line here is higher. And this is the, where the resistant cells are. So a higher dosage of the cytotoxic drug seems to select for more resistant cells after a period by which you don't really see anything happening. So in other words, you can think about this as a period of remission after which cells emerge, but with a higher level of resistance. And if you give an even higher dosage, you get a longer remission, but when the cells emerge, they emerge with a higher level of resistance. And you can go and compare. This is when the cytotoxic drug is given continuously. You can compare between that and what happens when you do it with an on and off administration. And I mean, this is not very clear the way I'm presenting it, but you may be able to see. And when I say on and off is you pick a period and you say in a continuous administration, that's the amount of drug I would have given you. Now let's give the same amount of drug in a shorter burst and do it in bursts. Metronomic. And what happens there with this model is that selects for even higher level of resistance.
Sorry, what's the difference between the red color and the theta variable?
No. So, the theta variable is just the resistance level, right? So what? So higher is more resistant. So it's some scaling that you do there too. Yeah, I'm trying to see. I do not have here. Oops, again, I'm pressing the. I did not put here one of the. It's basically some normalization to see where you get the maximum resistance. I mean, it is, yes, but yeah, unfortunately, I did not put here. I don't think that's sufficiently clear.
It's just that it's higher, the theta is higher.
Yes.
Okay.
Yeah. Well, the cells with a theta.
Right.
There are more cells with a higher theta. That's a way of reading it. Okay. Now you can study all these different contexts of what happens if you have cell diffusion or no cell diffusion. For example, here, without any cell diffusion, when you look at the no cell diffusion, but with a diffusion of nutrients, you see what happens when you increase the diffusion of the nutrients to the level of the resistance of the cells. And there are two scenarios here. There's a low drug diffusion. So there are four kinds of diffusion here. There's diffusion for drug 1, diffusion for drug 2. There's a diffusion for the nutrients and the diffusion for the cells. Here in this picture, no diffusion for the cells. There's an increased diffusion of nutrients goes from here to here, and increased diffusion of drug resistance goes from here to here. I would not place too much effort in these graphs, but just to tell you that, you can do all these things. What is actually more interesting in this case is the following picture. I think that is a good one. And what I'm showing here, it's not the resistance versus time. This is the resistance versus R. To remind you, this is still the radially symmetric case. We'll get to the non-radially symmetric case in a second. But this is the radially symmetric case. So what you see here is 0 is the very center of the tumor. 1 is the boundary of the tumor. And again, the vertical axis represents the resistance level. This is sensitive. This is resistant. So what you do here is you start from a certain level of resistance. Basically, it's all the time the same experiment. But under certain conditions, what you will get here is this very, very interesting picture. And let's think for a second about what this picture means. What that picture means is that inside the tumor, there is a certain level of resistance. Again, the x-axis is the radial variable, while away, there's a higher level of resistance. So what you see here is a scenario, and this was not the initial data. This emerged as a result of simulations of this model under certain parameter values. What you see here is something that corresponds to, if you wish, well, something that looks like that, but a radially symmetric version of it, right? So you have a region with lower resistance and a region with higher resistance that naturally emerged as a result of this mutation selection process. Okay, so this is interesting. The what? The x-axis is R. No, that's not the growth. Now we have space. So this is the distance from the center of the tumor. You're looking here at a radially symmetric case. So this is R equals 1. Okay, and what it says here is a certain level of resistance, low resistance, and here you have high resistance. And that emerged. Now that's a radially symmetric case. I'll show you how that works with the full 2D model, but in any event, that's a good example. You can also ask me what happens if the mutation, by the way, the mutation kernel was fully symmetric. What happens if there's an asymmetric mutation kernel? Well, that will just change the profiles. The red is with a symmetric kernel, the blue is with asymmetric kernel. The x-axis here is the trait from sensitive to resistant. So you can do that. In fact, you can go one step further and say, what happens if it's not even continuous, but there are different kinds of levels of resistance? You can also do that. There's no theorem behind that, but at least you can see what these pictures look like. So you have the model, you can play with it in many different ways. Okay. Given the radially symmetric case, the question is, how do I go now to a full 2D in space model? This is a little bit more difficult. And why is that? Because remember, in the radially symmetric case, the growth, I just said cells grow and cells die. But I didn't have to worry about where they grow and where they die. So once I do a real 2D, or if you want a 3D in space model, I actually need a model for tumor growth to embed it inside that whole framework. Now, look, I was thinking about this for a long time, and I realized that there is something strange in our community. That is, you look at the literature, and there's a lot of work that was done on models for tumor growth, and a lot of work that was done on models for drug resistance. but almost nothing that combines these two. And why? And the answer, I think, is obvious, and that is the dimensionality kills you. Okay, there is no reason why we cannot sit down and write these models, but to like simulate them, that is difficult. Proves things, and then, well, they become complex, right? So it's all complex, but look, a model that is 2D in space with one variable of resistance that's continuous and time, that's already a 3+1. If now I wanted to add more drugs, that increases the dimensionality and so on. If I want to do the 3D in space, so it's not any conceptual reason that prevented us from doing these things. It's just the actual mechanics of that made it difficult. So that's why you almost don't see works on that, unless it's like you see some of like the yes, no, resistance sensitive, but things in between, not that many. Okay, so you have to add something for the growth. So basically, full 2D problem will have a growth and death. So this G you see is here. G has a G, okay, here. Growth, natural death, and a death as a result of a cytotoxic drug. The cytostatic drug will still slow the growth, but the real thing is, and what's hiding here is really how to model the cancer growth. And what we did in this particular, and you can pick your favorite growth model, but we did in this particular case, we followed a work of Pertam where all that is happening here is there is a homeostatic pressure that pushes things from the inside. You have to nicely define the boundary. You take that thing and impose it on everything else. Then it's just details, but it's a model of your choice. This is a model of our choice. Okay, so now you have the model for tumor growth. Okay, and nothing interesting about the nutrients or the drugs other than, well, now that I have the 2D thing, before in the radially, no one asked me, how do I administer the drugs in the radially symmetric case or the nutrients? Well, the only way to do it is from the outside. But when you have, a radially symmetric case, so all that you're doing is you're providing the drugs here. If you have a magical way of putting it here, you can also do it here or here, anywhere you want, but that will be a bit silly. But in any event, you can administer the drug uniformly from the outside. That's how it was administered in the radially symmetric case. In the full 2D case, that already brings you different possibilities. And what I'm showing here are 3 examples. One is to administer it radially from the outside. The other is just uniformly say from the top. And the third, this was a nice paper of Mark Chaplin and collaborators where they did this simulated vasculature kind of idea. So you take this, blue regions and administer the drug or the nutrients through them and not through the reds. I mean, if you want an actual vasculature, you're welcome to add it to the picture. You can do that. Okay, so you do this, and the first thing that you ask, is that a good model for tumor growth? And by some measures it is. For example, what you see in this model is the emergence of a necrotic core. So over time, basically, you start from the beginning. I mean, like, I know I said I'm doing the 2D, but here is a radially symmetric case in 2D. Okay, so the tumor grows. As it grows, it's flat. And then all of a sudden, there is the emergence of dead cells in between. And you get this core of dead cells inside. You can do estimates on the velocity of the boundary if you wish, and all that stuff. In any event, it is a good model. So what do you do that? What do you do then? Well, you can repeat the same experiments, computational experiments that I showed you previously. So for example, what happens if you look at the evolution of the mean phenotype? Here, there is with a certain level of the cytotoxic drug. Here is with a higher level of a cytotoxic drug. And what you see for the mean phenotype is that there is a selection for something. The red means more resistant, something that's more resistant with the higher drug dosage. Okay. Here I did put the cross sections, so you can see what happens. with an increased level of drug. So this is a low level of drug. That will be the tumor on the boundary, and with a high level of drug, that will be inside. So you get a smaller tumor with a higher dose, makes sense, but more resistant cells. Now you can do it in many different ways. And what I'm showing here is a simulation where you start here with a tumor, quote unquote, red and blue. Red, fully resistant, blue, fully sensitive. And there are these 4 different regions here. It's something circular. And what I'm showing here are basically 2 things. One is I'm showing you the actual tumor. So that's the upper picture. And then inside is what is the resistance, level of resistance of the cells. This is kind of funny. That's the first time. So this is, it's rotated by 90 degrees, right? Because they're red and blues are like that. So that is not the initial data. That is the initial data rotated by 90 degrees. Okay, so this is with no drug. What do you see? The tumor just grows. Why does the tumor grow towards that area? Because we are diffusing the nutrients from the top. So the tumor will grow in that area. So it's not symmetric. What happens as the tumor grows over time? Well, you see for yourself. The resistant areas grow, the sensitive areas grow, the whole thing grows. Okay? Goes back to my initial question, what's the best we can do? Well, let's add some drug. So what happens if you add some drug? Well, yeah, you get a smaller tumor. But what happens with the resistance level? Well, I mean, you're left with very little sensitive cells and a lot of resistance. And of course, that is what you see if you add more drug. Here you can't really see it. It doesn't disappear, but okay, you're selecting for the resistant cells. Okay. Here is another example. And they're just like the same. This is correctly rotated. Okay, but fine. They're just separated blobs. Same story. Yeah, it looks, but it's not really. Yeah, it can't go away. There's nothing to kill that. I'm not sure. I don't know where it went here, but yeah, no, there's nothing to get rid of it in this. Once you select for the resist,
we're shrinking already.
Yeah, I don't.
20 to 30 hours, whatever.
Yeah,
I'm talking about the last.
Yeah, I don't. What? No, here. This is.
No, I mean at the bottom.
Yeah, I ignore that bottom. No, I don't like that. Yeah. No, there's nothing to kill. And you can also see, this is a little bit more like the picture that you will get a different region with different levels of heterogeneity while you do that. This is interesting, this one, where you do this simulated vasculature. What you get is something that actually looks like that with that level of the, like at the boundary. Okay. And I don't know if that's really meaningful or not, but it's interesting. The reason why it's interesting, first you see the level of resistance also over there, but the reason why it's interesting is because, the tumors, they come on the surface in different ways. So there's nothing here that you see. They can have individual cells, and they can have this diffusive sort of boundary, and they call this the different kinds of things that you see when you look at edges of tumors. So this is an experimental paper from 2008 where I took that figure from. And I mean, again, I'm not putting any big connections here, not making big connections, but this looks a little bit like that one. Yeah. So it's interesting that that's what's happening. I mean, that's all I'll say about this. Okay, you can play with optimal drug. So I have 5 minutes, right? I mean, yeah. You don't. Don't make me speak for 20 more minutes. Okay, so you can, there's one other, okay. I do not want to, I mean, like, okay, so I didn't really show you data, but what I want to, I want to skip to something else. One thing that is interesting here is, so I told you about, I said something about the curse of dimensionality. And yes, what happens if I have multiple drugs? So I can take here like several cytotoxic drugs, and I can take several cytostatic drugs. Now, I do not know how to measure their impact without someone telling me. The way things are written here is as if they're acting independently of each other. Okay, so the what happens to when you take several drugs that slow down the growth? How about just let's go and add their individual impact and ignore all cross-reactivity? What happens with the cytotoxic drug? Oh, let's just say that the probability that the cell dies is a probability that's not killed by any of these, and just have them act independently of each other. But that does not have to be the case, and it never is. So if you come with the experimental data that tells you how different drugs act in conjunction with each other, it is very straightforward to go and change these terms to something else that will encode this cross-reactivity. Well, in the absence of being able to do that, well, we wrote something that looks like that and studied it. And it was interesting because there was a paper of Gardner in JTB in 2002 that actually did not take a continuous trait, but had 3 states: a sensitive, a resistant, and something in between. So there are 3 points. And these are actually, there's experimental data that goes with it, but with 3 points. And it goes back to what I said previously. Like, we like big data. We like to train these machine learning algorithms on an enormous amount of data. Sometimes what we have is 3 points. Sometimes we have 2 points. I have cases with 1 point. Okay, no points is actually the common one, right? If you think about that. So no data is what you have really, but 3 points. Okay, but and there was a collection here of different drugs, and they're listed here as tamoxifen and Herceptin, so cytotoxic and cell cycle and non-cell cycle specific. But what was interesting now is you can go and study the dynamics of what happens when the trait is continuous. And yes, you have 3 data points, and you do see some differences and some similarities. I do not want to get into full analysis for that, but just to tell you that, you know, the continuum parameter captures significantly more, as you can probably expect to see. And finally, with the last 2 slides, what I want to say is that you can take it even one step further. And that is everything here, everything that I wrote was in vacuum. There are only cancer cells. You know, even the tumor is not just cancer cells. There are many other things that exist within the tumor. But think of the cancer tumor. Let's, you know, coin this as the same for the sake of this. But still, there's much more beyond. There's microenvironment. There is macroenvironment. There are healthy cells around. And one thing you can do as a toy problem is to take all that machinery and study it with the cancer cells growing in some environment. So a competition between, in this case, this N sub H, these are the healthy cells, and N sub C, these are the cancer cells. And there is a cross-reactivity terms here. They this is a term that involves both the cancer cells, their density, and the healthy cells, and this is the other way around. Other than that, it's the usual. It's the growth, the death, the cytotoxic drug, the diffusion, and same thing for both cells. But there are these reactivity terms. And there are different scenarios where you can study things. It could be like a low competition scenario and a high competition scenario, and you can do a simulation. So what I show here is the evolution of cancer cells and healthy cells, and it's a little bit interesting how things are prescribed, but like, you know, in the continuum, they really, these systems are written as if they actually live together at the same place. Okay, so we're not following here the membrane and say, well, this is a domain where the cancer cell lives and this is the domain where the healthy cells live, but we just assume that they're all together at the same time in the same place, and they're just competing with each other. And you get different patterns in terms of who, so this is over time, and the red cells are the cancer cells and the blue cells are the healthy cells, and who takes over and who wins. Okay, so you can study different scenarios, but all that I wanted to say is that things do not live in vacuum. So with that being said, let me conclude. I think that, I mean, I tried to be a little bit provocative with this science fiction vision, but it's more than provocation. It really is the fact that we are so busy doing the math sometimes and forget that, you know, not just Eduardo said, it's not just how do we convince the doctor that this is a value, but are we even asking the right questions mathematically? And I'm not sure we are, but I think we can start thinking and doing it in that direction. So with that, I'd like to thank you for your attention and end here. Thanks. Do you want to use a microphone? Maybe?
Yeah, sure.
I think this should.
Yeah, thank you, Doron. Very interested in modeling. So at the very beginning of your talk, like an hour ago, you talked about genetics versus epigenetics.
Yes.
I think your model now included genetics, right? A cell was born with a new trait, and then it would have the trait forever. Could you maybe also use this for epigenetics?
Yes, you can. This is not described here, but models that involve epigenetics in that spirit were recently constructed by Tommaso Lorenzi and his group. So that will be a place to look at, but absolutely, yes. The biggest issue, though, that I see with some of these models is the use of diffusion term with epigenetics, which I do not perfectly understand. So when diffusion and the trait with, yes, so that's the standard way of doing it, is a diffusion and the trait, and doing it with epigenetics and the encoding. I don't completely understand why that should be the case, but sometimes we put these terms in models because that's what we know how to do, or that's what we can do. But that's a place where if someone comes with a clever idea, that's certainly an opening. Yes. Jasmine.
Thanks for the nice talk. So I have, well, one question I have is, have you considered drugs like